Okay, I think we should get started. Good morning. My name is Ellen Leinfuss. I am an SVP at Certara Simcyp division, and I am joined today by our subject matter expert and speaker, Will Redfern. Today, we're going to be talking about Secondary Intelligence, which is an evidence-based secondary pharmacology software tool that we've developed to solve a very strong need in the marketplace. We've been working on this product for quite some time, and we're thrilled with how we've progressed. We're going to share that information with you this morning as a partner to SPS. Introducing Will Redfern, who is my colleague, is Vice President of Quantitative Systems Toxicology and Safety, which we abbreviate to QSTS at Certara. He is an experienced safety pharmacologist, having worked at Syntex, Quintiles, and AstraZeneca. Will has authored or co-authored over 40 papers, review articles, and book chapters. And Will is a fellow of the Royal Society of Biology and of the British Pharmacology Society. He's a diplomat in safety pharmacology and an honorary senior lecturer at the University of Manchester. He also received the Distinguished Service Award from SPS, this organization, in 2014, and also received from that organization its Technology Innovation Award in 2018. He is also a past president of the society. So with that, it is my pleasure to turn the stage over to Dr. Will Redfern. Take it away, Will. Thank you.
Thanks for the introduction, Ellen, and thank you everyone for dialing in. So yeah, I'm going to talk about this new software, Secondary Intelligence. So first of all, for those of you that are perhaps not familiar with Certara, you should be familiar with our products. So these have been used in over 90% of all new drug approvals by the FDA since 2014. So they include Phoenix, which is a PK/PD platform, Simcyp, which would be used by your DMPK colleagues, which is a PBPK simulator. We also have an in vitro data analysis toolkit called SIVA. We have a cardiac safety simulator, and also D360, which is a database and with functionality, which is used by several major pharma. So I want to talk to you about one of the QSTS tools that's in development, Secondary Intelligence. And this is part of Certara's ToxStudio concept. So ToxStudio, at the heart of this, is the core interface, knowledge and modeling integration, taking in vitro experimental data analysis and import modules, talking to modeling modules, importing other data, and outputting visualization and data output. So some of these Certara products that can interact with ToxStudio already exist. SIVA toolkit I mentioned, and also Simcyp Animal, which is obviously the animal version of the Simcyp engine. And today I want to focus on Secondary Intelligence software. So most pharma companies screen their compounds of interest against a broad panel of off-target receptors, and they spend anything from $500,000 to $1 million on this. And our question is best use made of the data? The readouts can potentially be used both prospectively to predict outcomes in non-clinical and clinical studies, and retrospectively to explain findings in non-clinical and clinical studies. Regulatory agencies ask questions on the profiling data, and there's been a growing prominence of this discipline over the last decade. So these are some of the papers, review articles from pharma. There's a couple here that were from single companies. So this one from AbbVie, and also this fairly recent one from Roche. And the others are intercompany review articles. And also from regulatory agencies, including one that's in press in JPTM, which is well worth a read. So certainly the effort going into, by opinion leaders in the field shows this is really an important part of our armory in safety pharmacology. When we gave a webinar on secondary intelligence in July, we ran a live survey, and I'd just like to reflect on the outcomes. So we had some people who dialed on the call who weren't doing secondary pharmacology per se, so they end up in the not applicable category. But for the first question, what data do you generate for regulatory submission? Most people did actually generate an IC50 or Ki and functional assays, so not just percent inhibition. They went through to IC50 or Ki and found out whether the compound was an agonist or an antagonist. Most people were using the data prospectively, trying to predict outcomes in the safety pharmacology and clinical development studies. And then our third question was, when it comes to the point of regulatory submission, how large is your secondary pharmacology screening panel? So it was a kind of a bit of a split here, but the majority were either using 50 to 75 or more than 100 targets. So that just shows that people are doing quite extensive profiling. At the core of everyone's panel, just about, is the Bose panel. And this was a consensus panel from AZ, GSK, Novartis, and Pfizer, where they agreed on 44 receptors, comprising 24 GPCRs, 8 ion channels, 6 enzymes, 3 transporters, and 2 nuclear hormone receptors and a kinase. And these were selected based on hit rate, or average hit rate, and the safety implications of hitting them. So it was an obvious starting point for us to focus on. So we adopted and applied a consistent approach across all 44 receptors in the Bose panel and put it into a user-friendly software package. For each receptor in the Bose panel, we expertly curated lists of reference drugs. So these are defined as drugs that target that receptor selectively for their therapeutic efficacy. So most of these are marketed drugs, but this also includes candidate drugs still in clinical development or discontinued drugs either in clinical development or withdrawn from the market. The main thing is there's clinical data available. So the data that we sourced and expertly reviewed were the Ki, or the IC50 at the receptor, the lowest effective plasma concentrations in humans, and plasma protein binding data in human. So there are 5 key aspects to the software: qualitative information, mechanistic insight, quantitative analysis, data visualization, and report generation. So the qualitative information, we basically list all the drugs targeting the receptor, including the ones that I've just defined. Also veterinary drugs and research tools. We list the main organs or organ systems affected, the commonest associated side effects, the liability of most concern if you have this as an off-target, expected safety pharmacology outcomes, and translatable biomarkers. In terms of mechanistic insight, we've constructed, where possible, mechanistic safety pathways for the most noteworthy side effects associated with the receptor interactions, each citing a useful paper review article in the mechanism. Quantitative analysis, this is at the heart of the software, and this really compares where your test compound sits in relation to drugs targeting the receptor for clinical efficacy. And then data visualization tools, looking at the data from all angles really, including multi-compound comparisons, and then generation of a report. So I'll cover each of these aspects when I give you a tour of the software. knowing the potential side effects from an off-target interaction is not enough. And what you really want to know is, how hard do I have to hit this receptor to see these side effects? Or from the other direction, how much engagement with this target can I get away with before I see these side effects? And this is going to vary between receptors and between pharmacodynamic effects of the drug at the same receptor, for example, in different tissues. It requires knowledge of how much target engagement is required by the marketed drugs targeting this receptor by evaluating the ratio of their effective plasma concentration unbound divided by the Ki or IC50 at the receptor, and then compare the predicted plasma Cmax unbound over Ki for your compound. Does it sit within the range of marketed drugs, or do you have a margin? So for each reference drug at a receptor, we sourced the lowest effective plasma concentration in human, the Ki at the receptor, and basically divided the plasma concentration unbound by the Ki. So when we've got several reference drugs, we plot these on a scale shown here. And where they sit on this scale, just to sort of walk you through this, where they sit on this line of unity, the effective unbound plasma concentration is equal to the Ki. If they're to the right of it, the free plasma concentration required for an effect is greater than the Ki. We haven't actually seen it this far out, but if it were to be right over to right, it's obviously much higher than the receptor Ki. On the other side, here it's the effective unbound plasma concentration is less than the Ki, and then out over to the left, much lower than the Ki. So here we'd start to question, are we taking data from a local delivery route, for example, inhalation, and looking at plasma concentration? So that would be incorrect to do. Also, does the drug have pharmacologically active metabolites? So if you're using the parent drug concentration, you're getting an erroneously low value. And I'll come on to that later in the talk. In fact, now. So I'm going to talk about pharmacologically active metabolites with an example for using lefepramine, which is a NET inhibitor, norepinephrine transporter inhibitor. So here, this point circled in red is the what you'd get with the parent drug alone. So it's giving you the idea that you only need a fraction of occupancy to get an effect. Whereas in fact, this is where the metabolite, the pharmacologically active metabolite is. And if you do a combined pharmacologically active analysis, you end up with a point there. So the point with the purple circle is obviously in a quite different position from the red circle. And that's the purple one is the one we would use for the analysis. This takes into account the activity of the metabolite. It's not as straightforward as it first appears, because to enable to do this, you need to source the Ki plasma protein binding and simultaneous plasma concentration of the pharmacologically active metabolite. And if any of this information is missing, we can't use this reference drug. The eventual position of this combination data point depends on the relative potency of the parent and the PAM at the target, the plasma concentrations of the parent and the PAM, and the plasma protein binding of the two molecules. The impact on slow and fast metabolizers will vary between drugs according to the above parameters. And we've encountered this in well over 25 reference drugs so far, and dealt with it. So I'm going to give you a tour of the software now. So these are screenshots from the actual software. I'm going to start with the home page, which is the project view. So this is where you're able to select or import a project. And all the project is a single Excel file containing receptor names and Ki values from your receptor panel readout. And the layout is simple and formatted for import into Secondary Intelligence. We're going to select this project called all-neg. So this is a fictitious compound which has a negative interaction, by which we mean it's either an antagonist at GPCRs, enzyme inhibitor, ion channel blocker, transporter inhibitor, negative interaction all across. So here we've clicked on all negative, and the current project is showing as this compound called all-neg. And that upload is achieved on a single click, and it's quite quick. So the next page, we're going to the project compounds view. It indicates the compounds that have been selected, all-neg. If there are multiple compounds, you can designate the focus compound to view. And I'm going to blow up this bit on the next slide so you can read it properly. But just note there's a scroll bar on the right that you can visualize more receptors than we can see on screen. So the receptors are listed in ascending numerical order of Ki values. In other words, the highest off-target affinity is at the top. In this case, it's acetylcholinesterase, and the Ki value is 0.1 micromolar. And then further receptors are visible by scrolling down. So this is one view you have, and this is effectively like the kind of readout you'd get from your receptor panel, which starts with the most potent interaction at the top, and then in descending order of potency. You'll notice that it's not a case that all the ones at the top are red, and that kind of gives the will actually reveal the fact that the occupancy required for different receptors for an effect varies between receptors. So it's not straightforward that you'll get red right at the top, and that's the key to the whole software and type of analysis. And then here on this panel on the left in the compound assessment view, the outcomes, red, amber, and green, are listed in order of, if you like, how far into the red each interaction is. So if you like, the most, the strongest interaction is with 5-HT1A at the top, and then it works its way down. And again, there's a scroll bar to take you down to the ones at the bottom are going to be amber and then green. And this plot on the right is just the Ki as each target. So the scale here isn't a ratio, it's the Ki or IC50 in micromolar. The vertical black line is your predicted free Cmax for the compound. So the most potent, as in that table, is acetylcholinesterase, and that was designated as red. And what we've got here is the diagonal cross indicates a negative interaction, as I mentioned earlier. And the color is high, medium, or low interaction, likelihood of interaction with that receptor in clinical use, which I'll explain in a minute. So there are different ways of visualizing the data. If you go then to the receptor panel view, this view here, which is laid out a bit like periodic table style, incorporates the Bose panel of 44 receptors, and then the symbols indicate whether it's got an agonist or antagonist interaction, or positive or negative, or not defined. And Not defined would be if you haven't done a functional assay, so you don't know what the interaction is. And then the color codes are low, medium, and high likelihood of interaction clinical use. So I'm going to blow this up in the next slide so you can see it more clearly. So on the left are the ion channels, and then the GPCRs, and on the right the enzymes and kinases, and here in the middle are the nuclear hormone receptors and transporters. So, obviously we put in a compound that had a negative interaction with all the receptors, this fictitious compound. The fuzzed out receptors here, VEGF2 kinase, are outside the Bose panel and are under construction, but kind of remained on the front page. So if you click on one of those receptors, sorry, what I meant to say was I'm going to click on the alpha-1A adrenoceptor. And this is, each receptor has its own page that looks like this. And in the top left panel is some information about side effects associated with the receptor. Then there's a list of drugs interacting with the receptor. There's a summary of the outcome. This panel on the top right is a kind of what if panel, and then there's the plot. So what I'm going to do is go through each of these one by one by blowing them up in a slide. If we start with the receptor summary, this contains useful information, including on side effects associated with interacting with this receptor, expected safety pharmacology outcomes, and translatable biomarkers. If the compound had a positive interaction, in other words, in this case, an agonist activity, then the plus column in the circle, this side, would have been highlighted and the other grayed out. So what we've got here is an antagonist interaction with alpha-1A adrenoceptors, and so the right-hand part of the table is highlighted and the left-hand grayed out. So the side effects, as you all know, the main organ system affected is the cardiovascular system. The liability of most concern, and this really means if you hit this as an off-target, is postural hypotension. Commonest associated side effects include dizziness, headache, nasal congestion, nausea, postural hypotension. Expected safety pharmacology outcomes are a decrease in blood pressure, and the translatable biomarker would be blood pressure as a measurement. Now, up in the top right-hand part of this screen is a button called Mechanistic Safety Pathways. So if you click on this, it takes you to this page. So we call these MSPs for short, and these have been constructed for each notable side effect where scientific knowledge permits it. And they use the format of AOPs, or adverse outcome pathways, that are used extensively in toxicology in the chemical and agrochemical industries. So if I click on one of these, which is on nasal congestion, just to blow it up so you can read it. So like an AOP, the left hand, the flow is from the simplest level of interaction, which in this case is with a receptor. So it's blocking a receptor, and there's the cellular response, the organ level response, and the organism response. So here it's saying in these little boxes, antagonism at post-junctional alpha-1A adrenoceptors on smooth muscle cells of specialized capacitance veins in nasal airway epithelium. When we were putting these together, we found the most interesting aspect was, where's the receptor? What cell type is it on? What tissue is it in? And often we might think of that in a slightly fuzzy way. So it actually obliged us to be very clear with ourselves. Where is this receptor that we're talking about? And then the next level up. So pharmacological antagonism of smooth muscle contractile response to noradrenaline, which is released continuously under sympathetic vasoconstrictor tone. and that the organ response, it reduces venous drainage from the nasal passages, swelling of veins in nasal airways, and then sensation of nasal congestion. And then there's a reference backing this up. So in a matter of seconds, really, to read that, you've got a very precise overview, which is consistent across all the side effects that we've done this for in all the receptors, in the same format, of how that comes about, rather than thinking, oh, finding a side effect of nasal congestion and thinking, how does it do that? What's that got to do with alpha-1 adrenoceptors? And it just saves so much time, and then it takes you to a reference, and then if you want to read more, you can, at least you've got a good starting point by chasing down that reference. If we go to the next panel, so this lists the drugs that target the receptor, in this case alpha-1 adrenoceptor, as antagonists. So drugs in bold are plotted as reference drugs in the plot I'm about to show you. M in brackets means marketed, W means withdrawn, D is a development compound, R is a research compound, and V is a veterinary drug. The links to the right in blue are to the IUFAR BPS receptor nomenclature guide, and Kemble also gives links for each drug or compound listed, and that's quite handy rather than having to go out of the software and you know, if you've got a link ready for IUFAR, fine, otherwise you're Googling it, so it's quite convenient. And then probably the core of the software is something that would take a group or one person probably, you know, several weeks to do for a receptor to do it properly. So this plot, as mentioned earlier, compares the plot of unbound plasma concentration of Ki for the test compound against a set of reference drugs. And also to point out that, as well as that, which is this scale here, above it is the margin scale, which is the inverse of that. So you might be more familiar. So this is kind of, you know, around a sort of 100-fold margin. So we've got everything covered. So this green, my cursor's red, but you can see the green circle plotted here with a diagonal cross. This is where your compound, or this compound all neg, has ended up. So in other words, it's deemed to be have a low likelihood of interacting with alpha-1 adrenoceptors in clinical use. If it had been in the red, then it would be assigned a high likelihood, and if it was amber, it would be a medium likelihood, and I'll come on to that shortly. And then this box, which was actually to the bottom left of the panel, the page, was a summary of the likelihood of interaction, and it allows you to enter a comment if you want to about the data or the compound. Now, at the top right of that page is what we call a what if panel. So this allows you to visualize what would happen if either the predicted Cmax unbound or the Ki for the compound of this receptor was altered, particularly useful for the predicted free Cmax. So for example, if you increase the unbound concentration by 10-fold, perhaps to reflect plasma concentrations to be seen in a phase 1 study, or maybe even if you're thinking about tox studies at the MTD. So here the scale factor's been increased to 10-fold from 1, and then you click apply, and then you can re-visualize the plot. So the green circle is where it was before, but this new symbol, which is now in the amber zone, is what happens if your actual predicted free Cmax is increased by 10-fold. So in this particular example, even if the unbound plasma concentration increased by 10-fold above the original value, the compound is still unlikely to engage with this target. It's in the amber zone, which I'll define shortly as how we would interpret that. But even pushing the concentration by a factor of 10 has not pushed it into the red zone. Just going back to some of those other data visualizations, the software can also compare interactions of multiple test compounds. We've got 4 shown here. So you can arrange these how you like. It gives you'd probably be comparing compounds from the same project. And we've got the ability to go up to 10 compounds. And the software will auto-generate a Word report, which you could use purely internally, or you could even append it to a report of a screening panel and form part of regulatory submission. And all the user has to do is to provide a conclusion paragraph. So all the text about the methodology, the tables, and the plots are auto-generated in the report. So I've mentioned this during the talk, and it's basically how do you interpret traffic light outcome. So here the compound has come out as green. And so the likelihood of interaction with this off-target receptor in clinical use, we would say, was low. And our suggested next steps could be, or would be, that further investigation is probably unnecessary. So in other words, if you get a green outcome, focus on something else, because this receptor is unlikely to be a problem as a side effect in clinical use. If it's in the amber zone, again, the advice would be investigate further if during the course of the project the predicted free Cmax increases. And if you go back to that what if scenario, you could actually look at how much you would have to increase before you got into problems with this receptor by the scale factor. If it's in the red zone, the suggestion would be to consider further investigation of the potential safety issue, either in vitro and or in vivo. So this could involve additional endpoints in safety pharmacology and or toxicology studies, or bespoke standalone studies. So the next question is, does it, this might sound quite sound, does it actually work? And the first point to make is that it would be impractical to attempt validation of each receptor interaction, which is not just 44, there's probably about 70, given there are agonist and antagonist interactions for most GPCRs. So what we are currently doing is taking a representative of each of the major classes of receptor in the Bose panel, an ion channel, the obvious one's hERG, which I'll come on to, a GPCR agonist interaction, and we've got to find one by trying to find which one has got drugs with off-target effects at that receptor. And in my talk tomorrow, I go into the issues around this sort of validation in more detail. A GPCR antagonist interaction. So I showed you the alpha-1 adrenoceptor page. We've got a couple of examples coming up. And then an enzyme. The enzyme we think will have most examples would be acetylcholinesterase. And then again, a transporter. We've got to try and find some examples for that. So I thought I'd just mention hERG. There are over 100 drugs listed in Credible Meds as having QT prolongation or TDP risk, which have the full data set required. So remember, you need hERG IC50, plasma protein binding, therapeutic plasma concentration range, and also a knowledge of whether there's pharmacologically active metabolites, and by pharmacologically active we mean at hERG. There are over 100 drugs that are not associated with QT prolongation or TDP, which have the full set of data. There is a lot of data to curate and check, so we're working our way through it, probably just over halfway through. Separately, there are also about 9 positives from thorough QT trial that we've got data for that have complete data sets, and numerous negatives. So we have 2 separate data sets achieving equivalent N numbers for positive and negative drugs. So the smaller data set from the thorough QT trial, and then a much larger data set where we're looking for whether QT is on the label. So, you know, present that at a later date. So alpha-1 adrenoceptors again. So as you may have noticed, we built the tool with 5 reference drugs: alfuzacin, prazosin, terazosin, tamsulosin, and celodicin. Doxazosin wasn't used to build the tool, but obviously it has alpha-1 adrenoceptor antagonism as its primary target. So it can be used as a validation drug. So think of the first group of 5 as the training set, if you like, and this is one from a test set. So move my cursor out of the way. So where doxazosin ended up was here. So it's in the red, thankfully. So it's correctly classified as red in secondary intelligence, which is reassuring. And it, you know, it's basically ended up in amongst its friends from the alpha-1 adrenoceptor antagonist club. Phentolamine is a non-selective alpha-1, should be alpha-1 adrenoceptor antagonist. It was not included, so can be used as a validation drug. And again, with phentolamine, it's ended up in the red. So again, that's reassuring that it's been correctly classified. So there's ongoing evaluation of other drugs with primary activity at alpha-1A. And also we will evaluate additional drugs with off-target alpha-1 adrenoceptor antagonist activity. And we thought we'd compare this with a previous publication from 2 of my former colleagues, Lynn Rosenbreyer-Ribeiro and Mark Petrus, who presented a poster at the Vancouver SPS in 2016. This was their data here, and this was the data plots for their drugs. And we looked at the drugs we've got in common, these 4 drugs, and basically there's a good agreement with the previous analysis of this receptor, which again is reassuring. So in terms of secondary intelligence in numbers, it addresses all 44 receptors in the Bose panel. We've evaluated over 700 reference drugs, from which 286 were plotted that had full data sets. We've constructed over 100 mechanistic safety pathways, and this shows the commonest associated side effects that we picked up for those interacting with those receptors. So slightly tongue-in-cheek, but this is a secondary pharmacologist life before and after secondary intelligence. So you get questioned on things like this fictitious project GHA. I see our favorite GHA compound has some antagonist activity at 5-HT3. Yes. What are the safety implications? I'll need to check our knowledge document and get back to you on that. What are the marketed drugs targeting 5-HT3? I'll just take a look at Wikipedia. But will it even reach plasma concentrations in the clinic that will interact with 5-HT3? that's really difficult to say. We'll have to compare it to the marketed drugs. This will take quite a while to do. Then after, same question, I see our favorite GHA compound has some antagonist activity at 5-HT3. Yes, but actually, what are the safety implications? Constipation is the main concern, but actually, what are the marketed drugs targeting 5-HT3? Allosetron, azacitron, celancitron, belasitron, all the cetrons. But will it even reach plasma concentrations in the clinic that will interact with 5-HT3? It's actually well into the green, so we shouldn't have to worry about that going forward. Why didn't you say that in the first place? So in summary, secondary intelligence contextualizes off-target hits at the 44 receptors in the Bose panel. And there are 3 ways of accessing the software. You can join our consortium, which will give you access to an additional receptors up to final total of about 200, plus other in silico Certara tools are ready now. And then other QSTS related tools will be developed, guided by consortium member needs. The second way to access it is to purchase an annual software license. And the third way is via consultancy projects, where we will run your compound through the tool and provide a report. So 3 things to think about in terms of different strategies. We'd love to speak to you, so you can arrange a call with us by contacting me. But before I go, I'd like to thank numerous colleagues who helped with the data curation and development of the software, who are listed here, and I'm happy to address any questions you might have. Thank you for listening.
Thank you, Will. We do have some questions, so have a sip of water, and here we go. Question number 1, first off, thank you, Will, for the nice overview. I was going to ask on the potential unbound plasma versus total concentration difference regarding, for example, intracell targets, but I assume that by entering 0% for protein binding, one can achieve a total concentration assessment. I'm going to read that again.
Yeah.
Okay. All right. So the question is on the potential unbound plasma versus total concentration difference regarding, for example, intracell targets. But I assume that by entering 0% for protein binding, one can achieve a total concentration assessment.
Yes, I think I follow that. But I think for intracellular concentration, if the receptor is like a nuclear hormone receptor, or the cardiac ion channels, particularly hERG, that you have to, the compound has to enter the cell to access, or any other intracellular target, I think you're going to have to think about other ways, perhaps using PBPK modeling to estimate what the intracellular free concentration of the compound is. But I think with what we're doing is if you like, the first step, and in some ways in many ways is a worst case scenario. So if you're already in the green, you don't have to worry further. If you're in the red, you might have to do some additional analysis if you want to try and dig it out of being in the red.
Okay, great. Second question. What did you do in the qualification of the content of the system to get it regulatory accepted? I'm going to read the second part here. As you know, science moves forward all the time. So how does Certara do to keep up things in date in annotating the new finding against targets? I think there's a two-part here. Yeah, I mean, I think just if I could just comment on the regulatory accepted part. I think we're in with this technology, secondary intelligence, we're in the early part of this journey, and we are just now introducing it from a regulatory perspective. Vis-a-vis Certara in general, Certara is very linked up with with gaining regulatory approval on all of our technologies. And we have, science teams that work continuously to see what's in the literature, to integrate that into all of our technology platforms, and to have a qualification verification loop. But I will turn it over to you, Will.
No, I think you've said it, Ellen, that, we've got to, the next stage of this is verification. And then talk to regulatory agencies to basically get their feedback on the technology. So, we're at that stage really. And what we're really claiming for this is it contextualizes your data. It's not a decision maker. And, you can end up with a compound in the red. And what you were really advising you to do is to follow that up. So if you decide to take the compound forward, from the software, you will know what safety pharmacology effects to look out for. So if they're already covered in a existing study type, you can, take note of them. If it requires bolting on some additional measurements into an existing study, then you can add those in. So for example, if it was blocking M3 receptors, muscarinic receptors, you might want to look at pupil size measurements that you wouldn't normally do, that you could easily do in a standard study. So it's really sort of pointing in the direction of what to look out for, and then you can make an integrated assessment. But the main thing is it gives you an early warning of what you've got in store further down the line. Eventually, secondary pharmacology data is trumped by in vivo and clinical data. So it's really, to give you an early warning of what's likely to happen. And then retrospectively as well, if you've seen a finding, this analysis can help you decide which receptor or receptors might be responsible for it.
Great, thank you. Okay, next question. At what stage in the drug synthesis safety screening would this assay best fit?
Well, as soon as you've got screening data. So some companies do a kind of a staged strategy where they might start, say, with 20 targets early in lead optimization, and then step it up to maybe 80, and then on the single candidate drug in at the time of the GLP safety pharmacology studies, you might go up to 100, 150, 200 receptors. So at all stages, but really it could be used quite early on once you starting to generate secondary pharmacology data.
Great. Okay, here's a good one. How would you handle compounds that have several off targets, some of which could have opposite effects? How would you assess what that combination effect could be?
Yeah, this is a good question. One thing that we thought of doing in a subsequent version, not really from this one, is a sort of simple algorithm that if you've got three, let's say if you've got a single target that results in increased heart rate, you kind of get a score for that. But if you're hitting three targets in, if you like, in the red that are associated with increased heart rate, I think you've got a stronger signal. The questioner is getting at if you've hit, let's say, muscarinic M2 receptors as an antagonist, but you've also hit beta-1 adrenoceptors as an antagonist. You're looking at heart rate, that can cancel itself out. We haven't put any quantitative quantitative analysis to account for this sort of interaction. It's almost by analogy like the SIPA model or cardiac modeling of different ion channel interactions, looking at effects on action potential duration, but rolled out for everything else. I think manually you can eyeball this, and you can make even with the software as it currently is, you can then see how effects might cancel out. But generally speaking, you know, we're talking about parameters that can change in either direction, and there's quite a lot that don't. So I'm trying to sort of think of other examples away from the cardiovascular system, but you'd have to sort of envisage things like seizure, where you've got a pro-seizure mechanism and an anticonvulsant mechanism via different receptors that are cancelling each other out. I think it gets very complicated, but manually at least you can eyeball the readout. You can look at the red interactions, and if you've got, I don't know, 7 of them, it won't take you too long to kind of figure out which ones might cancel each other out. But we haven't got that in the current software to give you a kind of a, feel like a arithmetical summation of the effects.
Great. And as the audience knows, Certara in SimSip technology is very much about combination. So that's certainly an area that we're working with in our quantitative systems pharmacology area is in combination therapies, combinations of different drugs, and what would their, what are the best outcomes and which ones to proceed with. So certainly this is, part of our DNA, and it will ultimately be part of this technology as well. Maybe 1 or 2 others. So let me give you kind of a softball here. What data do I need on my compound to enter into the software?
You need the KI or IC50, depending on the assay. In terms of for each receptor, you need to know the predicted free Cmax in clinical use, or in where you're going to go up to MTD in phase 1, in first in human. And you need to know the plasma protein binding. That's the only data you need.
Do you need to know if it's an agonist or an antagonist?
Yes. Now the software will actually work if you don't, and it will give you, can give you both outcomes. And really then it's quite interesting, because if you don't know if it's an agonist or an antagonist, one of the outcomes might be red and the other might be green. And then it becomes quite a pressure on you to actually go ahead and do a functional assay, because whichever way it comes out is going to be quite different. So I know that strategies vary between companies, and some companies automatically generate functional data at the same time they do the radioligand binding, and I think others will go back and do the functional activity if they get in a potent enough hit. So if you're at that stage where you don't know if it's an agonist or an antagonist, the software still will give you readouts, but you basically then have to decide, if you're getting green on both, then you don't have to bother. But if one's red and the other's green, then I think you need to go back and do a functional assay.
Okay, and then I'm just going to ask one other question, which I think you addressed earlier, but this company uses a larger panel than the Bose panel. What about other receptors, and what directionally, where are you going?
Yeah, well, obviously the Bose panel was the starting point, because it's common to larger panels, more or less. What I would say is that the Bose panel itself was selected on the basis of hit rate and importance of the off-target interactions. So from a hit rate perspective, this panel should serve you well most of the time. Most of the time if you hit something, it'll be in this panel. But our aim is to extend it beyond the Bose panel, stepping up, in increments of bundles of receptors to keep going. And, depending on customer demand, really, what people would like us to move on to going forward. But yeah, we are planning to keep going, but we had to start somewhere, and I think that the fact this is a consensus panel which will serve you well most of the time is a really good starting point.
Great. Well, thank you very much, Will. I'm going to conclude today's webinar, which is being recorded, and so it'll be available on demand. Thank all of you for coming this morning. I hope the rest of the day at SPS or wherever you're spending